Namaste friends, welcome you all for the biology session. In today's class, let us discuss about cell cycle and cell division. And this is MGS here from CET Sankalp crash course. So as we all know that cell is the fundamental unit of life. So what is cell? That is the fundamental unit of life. So fundamental unit of life and each and every cell in which the chemical molecules will be there and organelles like that organelles and the chemical molecules which interact, chemical molecules or chemical substances which interact, and that maintains the living condition of the cell. So when one cell can maintain its living condition, right, so that it can interact with many other cells, and as we all know that cells group together to form tissue group together to form the organ group together to form tissues, group together to form organs, and organs group together to form organ system. So if no cell maintain its living condition, no tissue, no organ system, and at the end no organism. Adike cells are the fundamental unit of life. So what is cell cycle? We know that cell division, where they are going to divide to form their progeny, or you can say that daughter cells. So what is it? Cell cycle exactly. So if you want to define the cell cycle, we can say that it is a life cycle of a cell. Cell cycle is the life cycle of a cell. What is that life cycle? So as we all know that cell, which will going to, if you're going to consider living organism, it grows and attain growth will be there and attain maturity, and attain maturity it gets a capability to reproduce, and it will going to produce a progeny. That is what it will going to continue its generation. Same like that, produce a progeny, right? Same like that, here we can say life cycle of a cell, in which the cell which will going to show cell growth will be there, and along with that where it performs its function, cell growth, and perform its functions, and after that here we can say that it will going to replicate, perform its functions. Replicate means duplicates. That is what it, after attaining maturity, it is ready to replicate, and after replication finally we can say that cell will going to divide. What is that replication? Means here it replicates the genetic material. So now we can define what is exactly the life cycle of a cell, or you can say that the cell cycle, it is the sequence of events. Sequence of events where cell will going to the duplicate events, sequence of events by which a cell's duplicates or cell duplicates its genome. I already mentioned that its genetic material, we can also call it as genome. Its genome, and synthesizes other cell organelles. Means duplicates not only genome, it also duplicates cell organelles, or I can say that the constituents of the organelles, and eventually divides. Once all those organelles will going to divide or duplicate, the cell is ready to divide. Eventually divides into two daughter cells. So this is what the definition of cell cycle, into two daughter cells. So what are the phases of cell cycle? So the phases of the cell cycle, right? For each and every cell, in general we can say there are two phases. One is interphase, other one is M phase. interphase and M phase. So here M phase in case of somatic cells, M phase in case of somatic cells, which is called as mitotic phase. Mitotic phase in the somatic cells. Somatic cells are also called as in plants vegetative cells in the Karitivi. So these vegetative cells are somatic cells which show division, right? So that is mitotic phase. And in sex cells, or you can say that germ mother cells, or what is it? Germ mother cells or gamete mother cells. Germ cells, not germ mother cells, sorry, gamete mother cells, or we can also call it as germ cells. So which will going to divide also M phase. Here M phase will be called it as meiotic phase. So interphase is common in both. Interphase is common in both. What is that? Vegetative cell or somatic cell and germ cells. Whereas M phase, mitotic phase in somatic cells, meiotic phase in sex cells or gamete mother cells. Let us take what is the difference between these two phases. Let us take first interphase. So interphase, which is inactive phase, or we can say that non-dividing phase, that is resting phase, or we can also call it as non-dividing phase. So what is that non-dividing phase? Where no, what is it, the division of DNA, or you can say that the duplication of DNA will be there, but no, what is it, the division of cell. The cells won't divides to give two daughter cells. So we can say that non-dividing phase, but in resting phase cells we can't say that inactive, that is metabolically active. Metabolically active means inactive in case of dividing condition. So they won't divide. And M phase, we can say here M phase in both somatic cells and sex cells, that is what is that, the meiotic division, mitotic division. So that is where you can say that nucleus will going to divide. And dividing phase, not only nucleus, what is that, both nuclear division, it includes a nuclear division and at the same time cytoplasmic division. So nuclear division and cytoplasmic division will be there. So yeah, let us take what is it, the difference between interphase and different stages, right? So here we can write, I will write what exactly the interphase which includes. Interphase is a common phase in both the cells, or you can say in the cell cycle. This includes G1 phase, which is called as gap 1 phase, and S phase, that is synthetic phase or synthesis phase, right? So we can say that the synthesis phase anta kuda karibodu, athwa synthetic DNA, that duplication phase, synthetic phase, replication phase, or we can say that DNA duplication phase. Synthesis phase, where DNA synthesis will be takes place, DNA duplication occur. And G2, that is gap 2 phase. So what is the difference between this G1, S and G2, right? Before going to know about, and M phase, I've already mentioned that mitotic phase in body cells, so where cells divides equally, and meiotic phase, that is where you can say reductional division. So here we'll consider that common, the phases which will be there. I already mentioned that, which includes a nuclear division, which is called as karyokinesis, and the one which includes, that is cytokinesis. What is it? Karyokinesis. Karyon means nucleus, so nuclear division. Cytokinesis means cytoplasmic division. So what is that cytoplasmic division? The cell completely divides. After once a DNA nuclear content of a cell divides, and then cytoplasm divides and gives rise to. So nuclear division or karyokinesis, which has generally 4 phases. So what are those 4 phases? That is prophase, metaphase, Anaphase and Telophase. Telophase. So prophase, metaphase, anaphase, telophase. So and finally after telophase, at the same time where the telophase takes place, that particular division, let us take what is a characteristic feature of telophase. Simultaneously there will be cytoplasmic division and finally gives rise to two cells, right? So next we can take difference between these G1 S and G2 phase. So I will mention here the interphase is the phase which occurs between what is it, two successive cell division, right? Interphase which is what is it, the present or occurs between the interval between two successive cell cycle. Interphase is an interval between a phase which is present between two successive cell cycle. Let us take what is the meaning of that successive cell cycle, right? So here we can take this one graph representation. So there I am going to represent what is it here. If you consider that this is a total 95% of the duration which takes place for interphase. So this which includes completely it is the interphase, right? So here we can write that is interphase, and here this includes the G1 phase, that is gap 1 phase, and here it is S phase and G2 phase. And karyokinesis and cytokinesis, that is what M phase, which includes prophase, metaphase, anaphase and telophase. Finally here we can say cytokinesis. So these are the phases. So let us take one by one. Sir, we're going to ask that question, which is the longest phase in the cell cycle? That is the interphase. That which we're going to take. Let us take point by point. So we can take this point. That is, it includes the series of changes in the interphase where cell prepare for what is it, the cell division. Preparation for cell division will be there in the interphase. Preparation for cell division will be there. What are those preparations? That cell has to grow, that is growth will be there, and DNA duplication, mainly DNA duplication occurs. So if DNA has to undergo duplication, some proteins and RNA is required. We can say that for that raw materials required, those raw materials will going to what is that takes place in this phase. So it is the longest phase, CET question, longest phase of the cell cycle where 95% of the total duration, so 95% of the what is that, the duration that takes place to complete this phase. So 95% of the total what is that, the duration of cell cycle which will be what is it, the occupied by this interphase. So cell cycle, 95% of the duration of cell cycle, what is that is why it is called as longest phase, and it includes three phases, G1, S and G2 phase. So what is that G1 phase? So let us take one here. I will write those things, very important thing in G1 phase. So G1 phase, cell grows and prepares for what is it, the DNA replication, same thing, and cell is metabolically active and lot of proteins and RNA will going to synthesize. Protein and RNA will going to synthesize, and nucleotides for the synthesis of DNA. Along with that, amino acids will going to synthesize. So what is it exactly, the amino acids? Amino acid required for the, the building blocks of proteins. Amino acids required for stone formation, or you can say that the production of histone or synthesis of histone. So keep it in mind that amino acids which are required for synthesis of histone, they will be producing G1 phase. But histone synthesis occurs here. Raw material ಬೇಕಾಗಿರೋದು G1 ಅಲ್ಲಿ synthesis ಆಗುತ್ತೆ. So utilizing that, what is it, the raw materials, that is amino acids, right, lysine and arginine residues will be utilized for the synthesis of histone protein. So histone protein role, that helps in the packaging of DNA material. So it helps in, what is it, the packaging of DNA, right? So that is what you have to keep it in mind. So we'll discuss that in the molecular biology. So, and in this, the final thing is that almost all the energy rich compounds are formed but not histone. Note, almost all the energy, what is it, the rich compounds, almost all energy rich compounds will be formed. So these energy rich compounds, but not, so I can write here, but no histone. So here histone in the S phase, histone synthesis takes place. So now here in this S phase, what happens? Let us take. So S phase, usually we know that there is a synthetic phase or DNA synthesis occurs. So one thing is that DNA synthesis. Among those G1, S and G2, this is the longest phase. So this, in interphase, longest phase is S phase. DNA synthesis occurs and it is a longest phase in the interphase. So there will be, what is it, the cell will going to, right, increases or double, right? So amount of DNA, that is where we can say like this. So amount of DNA, what is it, doubles. Amount of DNA doubles. The amount of DNA, keep in mind that per cell, in each and every cell which will going to take place, amount of DNA per cell doubles. And whereas no chromosome number increases. So point to be noted, no chromosomes, what is it, no chromosome number increases. So volume, or you can say that the quantity increases. DNA, if you consider 2C is a quantity, becomes 4C. Right? Not the number, I'm saying content. 2 content become 4 content, but always there will be 23 pairs of chromosomes. So chromosome number will be always same. So next we can take the G2 phase. So in S phase, we know that no, what is it, the number of chromosome won't change. No change in chromosome number, but there is a change in, what is it, amount of DNA. So next is here the third phase. This is a B and C, G2 phase. So gap 2 phase, what happens? Almost all the here cell continues to grow further and prepares for the division. So here we can say this is pre-mitotic division. So pre-mitotic phase, or you can say that pre-mitotic phase, because before mitosis. So it is a rapid, what is it, the almost all, there will be a rapid growth, and almost all proteins, here we can say all proteins and RNA synthesis will be takes place. So synthesize, and there will be a rapid growth. So rapid cell growth will be there. Now this is cell ready to, what is it, enter M phase. So here as it enters the M phase, mitotic phase, the first phase which is called as prophase. So in M phase, we're going to take, that is in case of mitosis, that is a mitotic phase, or mitosis phase or mitotic phase. So what is exactly here? Let us take point by point. So what happens in case of prophase and what is exactly? So here we can say that after this M phase, so once again here we can say after M phase the cycle repeats. So this is S phase. So in between, or you can say interval between 2 successive cell cycle. So here cell enters, what is it, the karyokinesis, and these, what is it, the 4 steps which are, what is it, the karyokinesis, and this is cytokinesis, and finally the two cells will going to produce here. Two daughter cells will going to produce. So here one cell which enters the cycle, and that will going to show G1 and then S and then G2. After that, those phases of what is it, mitotic phase, right? So let us take the characteristic feature. Before going to take that characteristic feature, right, we can say here end of the mitotic phase, two daughter cells will be produced. Two daughter cells will be produced. So where those two daughter cells which will be produced, the chromosome number is same. Chromosome number will be same. So, and as a chromosome number is same and two daughter cells, the number of what is it, cell increases. That is why it is called as equational division. Mitotic division is also called as equational division. So equational cell division, because it will going to what is it, the divide and produce what is it, two cells. So let us take one by one what happens in case of prophase, metaphase and anaphase. So I will write here first prophase. So in short form we can say P phase. So prophase, and what happens here with the first stage, right? First stage, right, where exactly it follows what is it, the S, right, synthetic phase and G2 phase. So next to G2 phase. So next to the G2 phase. Next to the G2 phase this takes place, and now here in the prophase, initiation of condensation of chromosomal what is that, material. So here first point, condensation of chromosomes. Condensation of chromosomes occurs. Condensation of chromosomes occurs. So initiation of condensation of chromosome. And second point, what happens here, we can say if you take this cell, and here the condensation starts. So before that cell, I will write here the nuclear membrane, right? So here I will take only nuclear membrane, it will be like this, right? So that is chromatin fiber. So now it starts and it will be appear like this. So where condensation occurs and centriole begins to move towards opposite pole. So in interphase, duplication of all those organelles occurs, and there will be two what is it, the centriole, and they are going to move towards the opposite pole. So that is why we can say centriole what is that, starts moving towards the opposite pole. Centriole move right towards opposite pole. Move towards opposite pole. So when they move towards opposite poles, right, so here move towards the opposite poles of the cell, and what we can say here, the condensation of what is it, the chromosomes occurs, and at when the chromosomes condensed, we can see chromatids appear. So chromatids appear. So chromatids appears, and their nuclear membrane, that is what we can say nuclear envelope, and then Golgi complex, mitochondria, all those cells which are going to disappear, disintegrate, or what is that, disappears. We can say those cells or cell organelles will going to what is that, disappear or disintegrates. Disappear or disintegrates, degenerates also we can say, right? So when nuclear membrane and all those things which will going to what is it, the disappear, we can say Golgi complex, endoplasmic reticulum, nucleolus, what is it, the nuclear envelope, that is what nuclear membrane. We can also say that nuclear membrane, all these will going to degenerate and produces. So now once this no boundary, the cells which will going to what is it, the spread. So complete condensation occurs. Condensation completes in the next phase, that is metaphase. So here these will be going to appear like this. So and these which will be there will going to spread throughout the cytoplasm. So as these chromosomes, condensed chromosomes spread throughout the what is that spreads throughout cytoplasm, right? If this is the cytoplasm, now what happens in the metaphase? So complete disintegration, which leads to what is it? The spread of chromosomes in the cytoplasm, throughout in the cytoplasm of the cell, spreads, or you can say that they're going to what is it? They spread throughout the cell. As these chromosomes are spread what is it? Through this cytoplasm of the cell, right? So that there we can say that the cell which will going to appear like this. So in this metaphase we can say that chromatids what is it? Clearly visible, where you can say that the complete condensation, so condensation of chromosome completes, chromosomes completes. As condensation completes what? So here we can say that the chromatids which will be there, and that can be in which the chromatids of the chromosome can be clearly visible. So here two chromatids are there, so these two are sister chromatids. This is a one chromatid, second chromatid, and two chromatids of the same chromosome which will be called as sister chromatid, and they are joined by centromere, attached by centromere, and there is a disc shaped structure which helps in receiving spindle fibers, that is kinetochore. So that is kinetochore. So now the condensation completes, and they can be, here we can say that these what is the structure what now I describe can be visible what is it? Under microscope. So chromosomes, right? Chromosomes can be visible under microscope. Hence morphology of the chromosome can be studied. Morphology of the chromosomes can be studied in what is it? M phase, that is mitotic phase, mainly in that karyokinesis, that is metaphase. So morphology of chromosomes can be studied, and I've already mentioned that there is small disc shaped structure on the surface of the centromere. There's a small disc shaped structure on the surface of the centromere. So if this is a centromere, right, on its surface which has disc shaped structure called as kinetochore. So this disc shaped structure which receives the what is that? Spindle fiber. That is why you can say what is the role of kinetochore if they're going to ask. Kinetochore serves as the site of attachment of spindle fibers. So what are these spindle fibers? Let us take that. So these spindle fibers which are generally formed by, so these spindle fibers which are formed by microtubules, formed by microtubules, right? Microtubules of the centriole, or you can say that centrosome. So these centrosome which are paired centriole, right? Paired centriole, and they're going to move towards the opposite pole, and as and when they're going to move completely to the opposite pole, if you consider this as a one centriole and this is the other centriole, and the chromosome will going to align in the metaphasic plate. How they're going to align? Let us take only two. Here the spindle fiber formed by microtubules of the centriole, right? So these spindle fibers which are formed by microtubules, right, of the centriole, and where they're going to attach to the centromere and passes through kinetochore attached to the centromere. So now wherever they will be, if you consider this diagram from here, right, so this spindle fiber will go and attached to this, now here it will go and attach to this, and it will go into constrict. Now it will pull to this region, and here from this pole, and this will going to pull this, right, or this one to the what is it, the center. So all those chromosomes with a respective end, they are going to pull and align in the metaphasic plate. So this is what is it, metaphasic plate. equatorial plate of the cell, equatorial plate of the metaphase, right? So that is what metaphasic plate, what we can say on this plane, or you can say that metaphasic plate is the plane of alignment of chromosome at metaphase, right? Metaphasic plate is plane of alignment, plane of alignment of the chromosomes, plane of alignment of chromosomes ಯಾವ ಫೇಸಲ್ಲಿದೆ, that is at metaphase, at metaphase, plane of alignment of chromosome, plane of alignment of chromosome at metaphase is called as metaphasic plate, metaphasic plate. So there the alignment takes place, and here the spindle fibers attach to kinetochore and pull those chromosomes and align into the equatorial plate. So here the characteristic feature is alignment of chromosomes on the equatorial plate, the equatorial plate. So after alignment, so that is where we can say exactly the chromatids can be. So those chromatids with the centromere, easily that structure can be studied. So next, so other one more we can say that from each, what is it, the pole, the spindle fibers which will go and attach with the chromosomes, and they're going to pull and align the chromosomes, right? So next, the third one, anaphase. So what happens in case of anaphase, as there will be the alignment of chromosomes which will be pulled by respective, what is it, the pole, and when they're going to constrict, the spindle fibers constrict, they're going to pull by breaking the centromere longitudinally. ಹಿಂಗಂದ್ರೆ ಕುಕ್ಕುಂಬರ್ ಪೀಸ್ ಎರಡು ಪೀಸ್ ಮಾಡ್ದಂಗೆ, ಒಂದು ಪೀಸ್ನ ಎರಡು ಪೀಸ್ ಮಾಡ್ದಂಗೆ ಲಾಂಗಿಟ್ಯೂಡನ್ಲಿ ಇಟ್ ವಿಲ್ ಗೋಯಿಂಗ್ ಟು ಡಿವೈಡ್. ಸೊ ಆ್ಯಸ್ ಇಟ್ ಡಿವೈಡ್ಸ್, ದೆನ್ ಈಚ್ ಕ್ರೋಮೋಸೋಮ್ ಗೆಟ್ ಸೆಪರೇಟ್ಸ್ ವಿತ್ ದೇರ್, what is it, the centromere, means centromere splits and with their sister chromatids. So that is they're going to move towards the opposite pole. So here when the constriction takes place, constriction of the spindle fiber takes place, and they're going to pull, what is it, sister chromatids towards the opposite pole. So this is a characteristic feature of the anaphase. So what happens, centromere splits longitudinally, centromere splits longitudinally. As they splits, chromatid separates. So chromatid, what chromatids, sister chromatids separates from chromosome, separates from each chromosome, from each chromosome. 23 ಪೇರು ಸೆಪರೇಟ್ ಆಗುತ್ತೆ from each chromosomes. So that is what you have to keep it in mind. That is, here you have to keep it in mind that the very important thing, they are going to move towards opposite pole. Chromatids separate and they are going to move, those chromatids move to the opposite pole, move to the opposite poles. And the last one here we can say that is the telophase. So what happens, once they start moving towards opposite pole, they completely move, right? So the chromatids, right, cluster at chromatids, one minute, chromatids, right, clusters at, right, opposite spindle poles, clusters at opposite spindle poles, we can say spindle poles. So when they cluster, right, so all those which were disappeared or disintegrated will going to reappear. So what is that, nuclear membrane or nuclear envelope assembles, right, and that will going to reappear, and nucleolus will going to reappear. So nucleolus, Golgi complex, endoplasmic reticulum, so reform. So reformation of all those organelles will be occurred in telophase, and at the same time cytokinesis initiates. So when cytokinesis initiates, right, so here we can say that exactly when this diagram we can write like this, this is a cleavage furrow in animal cell. So this is the cleavage furrow. It will keep on moving inward from this side. Just assume as this will going to keep on moving in, and finally they're going to divide. So once they divide, then two daughter cells at the same time, this nuclear envelope reappears, right, and chromatids will going to, what is that, decondense. Ones which were condensed, so they will going to, what is that, decondense. So this is very, very important, right? So at the same time, right, so which will going to divide. So karyokinesis, right, where cell in which the cytoplasm division takes place, right, together simultaneously, and telophase will be there, right, simultaneously this cytokinesis occurs. Cytokinesis occurs. For cytokinesis, cleavage furrow will going to help in animal cells, whereas in plant cells, cell plate formation. In plants, the division of there will be a cell wall, and cell plate formation will be there. So this is the thing you have to keep it in mind. So cytokinesis, which completes the mitotic phase, right? So in animals it is a cleavage furrow. In plants, what is that? That is cell plate formation. So cell plate formation, which are formed with the help of Golgi bodies. Golgi vesicles or Golgi bodies are involved here. Golgi bodies are involved, what is it, in the formation of cell plate. So that is why later that cell plate becomes, one more question we can expect, that later that cell plate becomes middle lamella. Middle lamella. So these are the things which are very important in case of mitosis. Let us take the significance of mitotic division. So once the cell which is going to divide, as we all know that, right, so in where exactly we can say that when each and every cell, right, which is going to be with their, what is it, the lifespan, once they're going to die, right, damaged cells, damaged or dead cells, which will be replaced by damaged or dead cells, right. It is not like that they won't replace, they're going to replace, right, through this mitotic division. We can say that in some case, if someone get hurt by, or you can say cut on the epidermal, the layer of the skin, where once the cell gets damaged, so damaged cell which will going to, they die. After that, what happens, that will be replaced by, the new cells. That a pre-existing cell will going to give rise to, if one cell which is there, that will going to give rise to two cells. So that is what the cell number, it helps to maintain the cell number and also volume, and finally there will be a growth. So finally growth. So growth and development is because of mitotic phase. So assume that if cell won't, do all those things, and there will be a error. If any error, in this case we can say, if any errors, right, if any errors, assume that no proper DNA and histone protein synthesis, or you can say that no nuclear division properly, once it completes the cytokinesis, cell which will going to arrest. So that is, the cell will be arrested. So that phase which is also called as quiescent phase. Cell arrest phase, which is also called as quiescent phase, means cell that do not divide further and exit G1, means as it has to enter after cytokinesis, it has to enter G1, but it won't enter the G1, it will going to leave that cell cycle, that is become G0 phase, which is also called as quiescent phase. G0 phase, which is also called as quiescent phase, and in that phase where metabolically active cell will be metabolically active, but it do not proliferate, means it won't divide until unless it will going to, get a message. Once that all those the errors set right, then only it will going to enter from G0 phase to the G1 and continue cell cycle, right. So this is the thing you have to keep in mind in case of What is that? So that is why cells do not divide. What is that? The further exit G1, right, to enter an inactive stage called G0. What is that? The phase. So these are the things. We'll take some questions related to this. So which one is correct? Which one is correct sequence of a cell? So correct sequence, as we all know that there will be a G1, right, ಇಲ್ಲೊಂದು ನಿಮೋನಿಕ್ ಕೊಡ್ತೀನಿ ನಾನು ಕನ್ನಡದಲ್ಲಿ ನಾವು ಹೇಳ್ತೀವಿ G1 name G1 send is friend G2, that is S. Here you can write S phase, send G1 send G2, that is we can say G2, right, to write PMAT exam, so that is what G1 S and G2 are interface, and PMAT, after that this is a nuclear division, and after that there is a cytokinesis. So these are the things you have to keep in mind. So G1 S G2 and G1 S G2 and M phase. So that is C is the answer. So next, given diagram, select what is it, the correct answer, what it represents, A B C D. So identify A B C D in the given diagram and depicting a cell cycle which is representing cell cycle. So here I already mentioned that if it's not enter this cycle, if it does not continue, that is a quiescent phase. So what is that? Quiescent or G0, cell arrest phase. So A is G0, so where we can say only in first option we have. Let us take next, continue, and B, if it is enters, that is G1 and then S phase and G2 phase, finally M phase. So if 24 hour cycle will be there in eukaryotes, cell cycle completes, right, in 24 hours. So generally 95% means approximately something around 23 hours which will be taken for interphase, to complete interphase. Only 1 hour, less than 1 hour for M phase, So let us take answer G0 phase. And here one more question we may get, the prokaryotes which will going to complete their life cycle, prokaryotes which complete that cell cycle within 90 minutes. 90 minutes means less than 1.5 hour. So that is why we can say the cell cycle, duration of cell cycle varies from organisms to organism, organisms to organism, or you can say that varies from organisms to organism, and also cell to cell. So this is what you have to keep it in mind. So each cell divides once in approximately every, what is that, 90 minutes. So then we give sometime 1.5 hour. So 1 hour 30 minutes is also correct. So 90 minutes it will going to finish its, what is that, the division. And which phase occupies a maximum part of a cell? As we all know that the 95% of the duration and the total what are given the diagram there, and this is occupied by interphase. So answer for that is interphase. So mitotic phase, that is in the somatic cells, I already mentioned this. Meiotic phase in, what is it, this 6 cells. Cytokinesis, division of cytoplasm. Next, which of the following phase of a cell cycle is not a part of interphase? So interphase includes G1, S and G2, whereas M phase, which is not a part of interphase. So one more thing here we can consider that. So once a cell cycle, if we can say, when cell which will going to divide, so after M phase, once again it enters G1 phase, right? So G1 phase is also called as post mitotic phase. Mitotic phase. So G1, yes, and G2. So G2 is pre-mitotic, before mitotic division. So pre-mitotic phase, and G1 is post-mitotic phase. So these are the things you have to keep it in mind. And most variable. So among these, here we can also write the duration. So 24-hour cycle, that is M phase, which takes less than 1 hour, and S phase, which will take some of, what is it, the out of 23 hours, it will take some 10 to 12 hours, right? And G2 phase, something around 4 to 6 hours, we can say 4 to 5 hours, and G1, which will be vary, that is 5 to 6 hours. So among this, so most variable phase, so G1 is most variable phase, right? So there will be, once if cell get arrest, so it takes more time to complete, right? So that particular G1 phase. So next question we can take, that is, in which stage DNA replication takes place. So as we all know that DNA replication which takes place in the S phase. DNA replication takes place in the, that is what, DNA replication which takes place in the S phase, hence it is called as DNA synthetic phase or DNA duplication phase. And we know that what happens in G1 and G2. So raw materials for histone will be produced here in the G1 and G2. Protein and RNA continues. Production of protein and RNA, metaphase, karyokinesis, and cytokinesis. Select the incorrect statement regarding S phase. It occurs between G1, S, and G, that is G1 and G2, that is correct. And DNA replication. So DNA replication, right, replicates in nucleus in this phase, that is also correct. Centriole duplicates in the cytoplasm, that is correct. So if it is a nuclear membrane, and here will be centriole, so duplication of centriole which will be takes place. And that centriole, when duplication occurs, it forms, that is the star shaped structure, that is astral rays. So that is what, when it starts the giving spindle fibers with the help of microtubules, that is astral rays, so asters. So that is what it looks like, what is it, the star, right? So that is correct. As DNA doubled, number of chromosome doubles. No, DNA doubled but not the number of chromosomes. So that is the incorrect statement is D. Incorrect statement is D. Cells that do not divide further exit G1 phase to enter an inactive stage, which is called as G0 phase, or which is also called as quiescent phase. Which is also called as quiescent phase ಅಂತ ಕರಿತೀವಿ. So where metabolically active but further proliferation won't be there, division. Which of the following statement is correct? Animals can show mitotic division in both haploid and diploid, what is it, the cells. So this question is, we can say that select the correct statement. It is not like that the mitotic division which takes place in haploid, what is it, the cells. So always in diploid, right, mitotic division in diploid cells, there will be meiotic division. So animals can show mitotic division in both diploid and haploid. So meiotic division will be there. In haploid, no mitotic division. Mitotic division won't be there. So that is not the correct statement. After S phase, the number of chromosomes become double, 2n to 4n. After S phase, the number of chromosomes become double. So that is, number won't change. Keep it in mind. Chromosome number remains same, but the chromosome content, volume increases. So during G2 phase, proteins are synthesized in preparation for mitosis while cell growth continues. So that is correct. So there will be protein synthesis will be there and cell growth continues. S or synthesis phase marks the period during which RNA synthesis takes place. No, it is not RNA synthesis, that is DNA synthesis. So correct statement is C, right? Which of the following stages a chromosome is minimum coiled. So generally in interphase where DNA duplication takes place, there is a minimum coil, and once it enters, the condensation starts, right? It promotes, or you can say that it will going to, what is it, they show little condensation. So condensation initiates in the M phase. So very minimum coiled will be there in interphase, where it enters, complete condensation will be there in the metaphase. So prophase, little condensation, and maximum slightly coiled condition, and maximum coiled, that is in the M phase, and then anaphase and telophase, that will be decondensed. So mitotic spindle is mainly composed of, so as I mentioned that these mitotic spindles which will be produced with the help of centriole, and those centrioles, right, those centrioles which helps in the formation of spindle fibers, and that spindle fiber, right, so that is what here you have to keep it in mind, that as a centriole which move towards the opposite pole, there they forms astral rays, you can say like this, spindle fibers which forms astral rays, and along with that, so once the astral rays which will be there, and centriole pair, so astral rays and centriole pair, like this it will be, like this it will be, and this is how, what is it looks before going to move towards the opposite pole. So that is what you can say, astral rays and centriole pair, so together which will be called it as aster, What are those astral rays? So when these, what is it, this centriole, when these centriole which is going to form spindle fibers, so microtubules, in simplest way we can say like this, microtubules, what are the microtubules of the astral rays, microtubules which are also called as astral rays of, what is it, the centrosome, which helps in the formation of spindle fibers, So microtubules which will be there, the protein called as tubulin, or you can say that tubulin protein which helps in the formation of spindle fibers. So when two, the centriole start moving towards the opposite pole, that is two asters, we can say two asters when start moving towards the opposite pole, spindle fiber formation occurs with the help of protein called as tubulin. So myosin and actin, these are the proteins present in the myofibrils, proteins of muscles, skeletal muscles, or you can say present in the myofibrils. Actomycin, both actin and myosin, that is which forms a cross bridge. So cross bridge in which both are, in association, forms actomyosin. Next, microtubules are absent in, so they will present in flagella, centriole and spindle fibers, but absent in mitochondria. So that is why the answer for that is mitochondria. In mitochondria, no microtubules. So direct question. So microtubules are with a protein called as tubulin. Utilizing the tubulin, spindle fibers originate, right? So that is what tubulin is, a microtubular protein. You can say like this, tubulin is microtubular protein, right, from which spindle fibers originates. Right? So these are the things you have to keep it in mind. So next, centrosome undergo duplication. Centrosome means paired centriole, so paired condition, undergo duplication, and that which will going to produce. Where exactly the duplication takes place? So generally we know that almost all the organelles duplication takes place in S phase. So one, it is S phase. In these two options we have, here no option. And what is it? S phase comes under interphase. So S phase comes under interphase. So here S phase and interphase in both we have, and begin to move towards opposite poles of a cell during. So when they're going to move towards the opposite pole, generally anaphase. So they start moving towards the anaphase. So if you consider that the anaphase which is going to takes place, that is takes place during the M phase, that is a mitosis. So answer for that is A. Right, sorry, answer for that is B. So B is the answer. So next, dash is the best stage to count a number and study of morphology of chromosome. So we can exactly see those chromatids along with centromere. Chromatids plus what is it? This centromere, which will be attached. Those two chromatids attached with the help of centromere that can be studied in M phase. So dash is the characterized by all chromosomes coming to lie at the equator. Which phase? In which phase we can say that chromosomes coming to equator with one chromatid connected by kinetochore. Right, so kinetochore, I've already mentioned that what is kinetochore. It is a disc-shaped structure which surrounds the centromere, through which spindle fiber passes and gets attached with the centromere. So this is a centromere, and this disc-shaped structure is called as kinetochore, which receives, kinetochore is a disc-shaped structure which receives spindle fibers which are formed by microtubules of the centriole and attach and helps in the alignment. So spindle fibers from one pole and its sister chromatid is connected by its kinetochore to spindle fibers formed from the opposite pole. So means from one side, if this is the chromatid 1 and this is one more and this is the centromere, this place is centromere and this is the kinetochore, to which from one pole, from each pole, that is spindle folds we can say, from one pole to the one kinetochore and opposite side, and from the other, what is it, the spindle pole to the kinetochore, so attachment, so in which will the characteristic of what is that, metaphase. Read the following statement. In mitotic cell division, chromosome number is halved. Right, so it is wrong. That is double. Chromosome number, which will be not halved, sorry, maintained, constant. Number remains same. Number remains same. When number remains same, here we can say that is what is it, read the following statement and which of the statements are correct. We have to select the correct statement. So this is wrong statement. And centromere is a point where two sister chromatids are held together. That is correct. The period between two successive mitotic division is known as telophase. Wrong. That is interphase. That is not a. That is interphase. The period or a phase between two successive mitotic division. So here the mitotic division starts and once it enters the interphase, and once again this is an interphase, and once again it enters the mitotic phase. So every time it will pass or it shows interphase. In G1 phase of cell cycle, protein and RNA are synthesized. So mainly where we can consider this, the protein and RNA which we are going to synthesize in both G1 and G2, but in G1 also there will be production, and this is a wrong statement. So that is second and fourth option are correct option. Second and fourth option only. So this is the thing. During karyokinesis, the spindle fibers get attached to condensing chromosome at highly differentiated region. That region is called as, during karyokinesis. What is the meaning of karyokinesis? That is nuclear division. Spindle fibers get attached to the condensing chromosomes at highly differentiated region. What is that highly differentiated region? We already discussed that. That is the disc-shaped structure. Which surrounds the centromere. Disc-shaped structure that which surrounds the centromere is called as kinetochore. So these are the things you have to keep keep in mind. Next, identify the structure indicated by what is it, the 1 to 4 indicated by, so 1 2 3 4 options. Let us take one by one. So what is that one which in what is it, the indicates generally in this case they've taken only, they've shown only one, so not both. So here we can take that clearly, and they are not indicated these two. If these two together, we could have select what is it, the chromosome, but only one they have pointed here, that is a chromatid. So one which is chromatid, and B, that is 2 which is given here, this point, where it is attached with a centriole, correct, right? So from each side there will be a centriole, so this is centriole 2, and 3 which was there, centromere, and finally we can say that, see how they have given completely, that is chromosome. One point matra hige kottidare. So that is, if you want to represent like this is the one, what it is, that is a, what is it, chromatid. So answer for that is chromatid, centriole, centromere and chromosome. So here we can select this one, chromosome wrong, chromatid correct, centromere correct, so sorry, centriole wrong, chromosome correct, sorry, chromosome wrong, right, centromere correct. So these options are wrong and A is the correct answer. Which of the following statement is not correct regarding colchition? So colchition is a chemical which is used to, what is it, the induce, right, so mitotic division further, and where it will going to duplicates, right, so duplication will be there, right, where it inhibits further replication, right. So that is what you can say, assembly of prevents assembly of microtubules is not correct. Okay, now it inhibits a chromosome replication. So replication will be there, but no division, means chromosome divides and they're going to increase their number, right here. So when they have to divide, right, when chromosome has to divide, and they're going to move, where they're going to inhibit the spindle fiber, they're going to, what is it, they affect the formation of spindle fibers, and it is an alkaloid and it is a mitotic poison, means no further division takes place. So that is what ploidy nature of a cell. See here also, that is the chromosome, here also if you consider 2n, there will be a 4n. So that is what ploidy nature increases. So to increase that, colchition will be taken, but it won't inhibit, it stimulates the chromosome replication. So incorrect statement is B. At which stage of mitosis the two daughter chromatids are separate from each other? Two daughter chromatids separate from each other. If you consider these are the daughter chromatids, right? So sister chromatids. At which stage of mitosis, they're asking, not meiosis, two daughter chromatids separate from each other and migrate towards opposite poles and now referred to as a chromosomes of future daughter. So that is what, now this is a metaphase and here they're going to divide. So and finally reach the, what is it, respective pole. So that is what in the anaphase. So these are the questions here you have to keep it in mind. So next further we can discuss in the next part. Thank you. Meiosis we can discuss in the next part.